And now it's time for our keynote lecture, and we're running a little behind. So I'm going to give a very short introduction and welcome Eileen Anderson, who is the director of the Sue and Bill Gross Stem Cell Center at UC Irvine. Eileen.
Okay, so I know, got it. I'm all that is standing between you and like a break and coffee, so I will do the best I can. I'm not sure how close I'll catch us up, but we can see what we can do. I wanted to give a little bit, a little bit of a non-traditional talk today. I figure people are probably getting tired, and it's data heavy, a lot of topic switching. So when I talked with Francesca about my presentation, I asked what she thought she would like to hear, and it was really the idea of a translational path. And so I'm giving more of a path level talk for what it means to try and move out of basic science and into the realm of translation. So it's a really broad scale overview of kind of one step forward, one step back, one step to the side, and hopefully a way that is encouraging for those of you who are earlier in your career to be able to think about, one failure is not the end of the world, and there are ways around this and to move forward. And also, I think, really to try and emphasize the importance of patient advocates in terms of helping us all to see the relevance of our work and really where we're going. And so kudos to the patient advocates, our speaker at the beginning, and the next one coming up, because I think that kind of interaction is just so important. So in this overview theme, I'm going to start out talking a little bit about how regulatory agencies view what we do in the context of stem cells. So regulatory agencies have the job of evaluating risk. They don't actually care that much about your basic science. They're all into the risk game. And cell origin and type influence risk, which all of you should know. Boy, this is delayed. There we go. So pluripotent cells that can make anything have one level of risk, right? The farther we go up this tree, of differentiation, right, up towards the branches and the leaves in our illustration of what a stem cell differentiation would be, towards multipotent cells or fully differentiated cells, those cells are going to have lower risk, right? And so everything that I'm telling you today is built around the cells that we have mainly worked with in the lab, which have a slightly lower risk than starting with the pluripotent stem cell population. These are tissue-educated cells that are derived from fetal tissue. Boy, this is wicked delayed. There we go. 16 to 20 weeks. Most of what I'm going to talk to you about is 16 weeks gestation. We've never seen these cells to form a tumor after transplantation in constitutively immunodeficient animals in any model that we've looked at. Oh, wow, now it's going way forward on its own. I apparently do not have sufficient patience for this device. Single-sorted cells form neurospheres in culture. They're multipotent. They retain that for many, many passages in a dish. And re-isolated single cells have the capacity to make new neurospheres, right, in a clonal assay. So they really are stem cells, but they're farther up the branches of that tree in differentiation. All right, I'm going to try and do this in like slow motion. There we go. So the other piece that I want to just lay out in terms of a translational path or timeline is that, as doubtless many of you have heard before, when we think about biologics and cell therapeutics, there are different issues that come into play in terms of thinking about preclinical milestones. And there's an interplay between our basic science and, if you work in a translationally oriented lab, how you bring that basic science forward into something that could be sort of reduced to practice, right, have applicability. And that comes into the domain of patient expectations and what happens for clinical trials. Patients in my area of research, spinal cord injury, have high hopes and less patience than I do even at this podium, and no real alternatives, right? And so that drives a particular mindset in people who might be coming to the table to volunteer as part of a clinical trial. Despite how far we've gotten in the last 20 years or so, these are novel indications, early experience in terms of moving things through to translation. And that matters in terms of what our realistic expectations are, even the questions that we know to ask, right? We have limits in terms of our animal models and the kinds of questions we can ask, how predictable human cell populations are going to be based on that data. We have to recognize that cells are going to have quite pleiotropic mechanisms, right? It isn't the case that we're going to replace, an enzyme outside of a hematopoietic genetic condition, right? For most of what we do, certainly in neurological disease and injury, we're transplanting cells with the expectation that they're not going to do just one thing. We have to presume that those transplants are going to be there for a long time, while some models have shown, in fact, that long-term persistence of the cells isn't necessary. We know for sure with these cells in spinal cord injury, if you lose the cells, you lose the biological effect, right? And so, in fact, we need them to be there long-term. We have to recognize that how well we control these cells in a dish or in vivo is really pretty poor. That's the reality. So when we're asking them to go someplace and do something in particular, we're not asking it very well, and they may not be listening to us very well. And lastly, and critically, because I'm going to spend some time on this later, we have potential for donor variation for these cell populations and batch-to-batch variation in terms of how we bring them forward that we don't understand there at all. And there's a lot of basic science that needs to be done in that space, I think, for us to be really effective as a field. Okay. So I'm going to give you kind of one timeline from my group for how this went, just to give a sense for folks in the audience who might be thinking that this is their career path, whether it's in academia or in industry, because I think that kind of perspective is sort of important to have. I didn't start out as a stem cell biologist. I was actually a bioengineer, and then I became a neurobiologist, and I started studying. I moved from Alzheimer's disease to spinal cord injury. I started my lab at UCI studying straight spinal cord injury and inflammation in 2001. And in that process, we developed a bunch of models that turned out to be really useful for thinking about doing cell transplants in. And so I was approached by some collaborators at colleague institutions and who had started a startup company saying, hey, would you try our cells in your model? At which point I started my path towards being a stem cell biologist. So in the first few years of that, sort of 2002 to 2005, we did a boatload of transplant studies, and we evolved really pretty much the first data to show that you could transplant neural stem cells and get recovery of function in an animal model of spinal cord injury. To this time in the spinal cord injury field, very few things had shown recovery of function in an animal model. So this was pretty exciting for us. Within 8 years of starting that work, that had actually gone forward to the first clinical trial for thoracic spinal cord injury with a neural stem cell transplant in a chronic condition, right? So only 2 trials had gone forward at that point for cell therapeutics for spinal cord injury. This was the only one that was in a more chronic condition. Over the subsequent years, 4 to 6 years after that, we really worked towards cervical as an indication because that's the majority of folks with spinal cord injury. It's by far the most devastating injury. And it has the advantage for us in terms of looking from a clinical trial perspective for repair at one level of recovery of function gives a really big impact, right? On the other hand, the risks of transplantation go through the roof compared to doing it thoracically. And so it's a give and take in terms of, you know, the pros and cons of which models you pick and which clinical indication one is targeting. We had a massive step back in 2017, which I'm going to come back to, where we came out and actually publicly disagreed with the company we had been working so closely with throughout this period. We were testing their final clinical product for cervical spinal cord injury clinical trial, and we didn't feel it had an adequate efficacy or safety profile. After a very public and unpleasant dispute, that company actually went under. They went out of business, and we were left trying to decide, were we going to continue work with this kind of stem cell and move forward along this path in my lab or do other things that we had going on in parallel? We ended up going forward, and I'm going to tell you a little bit about that story. There we go. I wanted to highlight just a little bit kind of the sense of how that funding works and really acknowledge that California Institute for Regenerative Medicine has been so pivotal for this kind of work in California. And you know, how stem cell research has advanced nationally in reality. It just isn't possible to do studies on this scale without the type of funding that comes from CIRM, even though we've supplemented that along the way with funding from National Institutes of Health and looked for ways to backstop, you know, our investigation, for example, of basic science mechanisms. CIRM is really a pivotal game in making this translational advance for what we're looking at for cell and gene therapies. So a shout out to them. Okay, So what did we know at this period as we were getting ready to think, wow, this is exciting data. What might really happen if we're able to go forward to a clinical trial? What you can see here, I just want to give you a sense of what this looks like in an animal model. You can see a vehicle on the left and a neural stem cell transplant on the right. You should be able to see that the animal on the left is mainly dragging their hind limbs, not able to step with their hind limbs. But the animal on the right is able to not just step with their hind limbs, but step in a coordinated manner with their forelimbs, right? So this isn't a small degree of functional gain in these animal models. It was a really substantive one that was exciting for the fields. Our key findings here with that in a subacute model, we got this really quite gratifying level of functional recovery. But anecdotally, we almost stopped this experiment before we had that data. We had planned to have these animals survive just 8 weeks, which we felt like was a really long period of time. Honest to God, my lead two technicians were out. I went in to do the animal evaluations on the weekend. The sacrifice of these animals was scheduled the next week. And I looked at them and I said, something is going, I'm blinded, of course. I see 2 different groups here. We need to prolong this experiment. We ended up having to go 16 weeks of engraftment. We now routinely do 24 weeks of engraftment because human cells and animal models can take a really long time, and you miss stuff if you don't go long enough in letting them engraft and migrate and differentiate and do their job. We saw this very robust engraftment into the host central nervous system. And we showed quite pivotally that if we ablated those human cells specifically using diphtheria toxin, we lost that functional recovery. And so we knew that sustained engraftment of these cells was going to be very critical. So that brought us through, as I already outlined a little bit, into switching towards cervical spinal cord injury after a pretty successful thoracic spinal cord injury trial. I want to give you a sense of what is involved in that work. So if you have a lab that works on basic science and translational science in parallel, this is the set of questions that we had to go to Swissmedic, to Health Canada, and to the FDA with. Replication of efficacy, transplant location, method, therapeutic window, mechanism of action, safety tox profile, immunosuppression and withdrawal of immunosuppression. Those all require different animal models. Dose and safety so that we could justify allometric scaling on the clinical side, and potency testing with the intended clinical cell lot and line. And that is the number of publications that came out of just doing that work of trying to move forward on a translational scale. And yet, we got to that threshold of going forward out of a thoracic model and into a cervical spinal cord injury model and had this full hard stop where we could not get efficacy with the intended clinical cell lot and line, and as I said, what ended up being a pretty public dispute in terms of those studies. That wasn't crazy, so we had to step back and evaluate a little bit. And a critical thing to understand that I think is worth talking about is this issue of potential donor variation for stem cell populations, and really importantly for batch-to-batch variation in how we bring those cells forward. So what do I really mean by that? A clinical study could employ, theoretically, more than one cell line and batch. The FDA doesn't preclude you from doing that. What they ask you for is to show comparability across different batches or across different lines. And that is almost always in vitro data. And this is where I think we have a challenge in the field because we just aren't good enough at what that in vitro data should be at this point. So an embryonic stem cell ESC here, for example, is almost always going to be the same line because of its expandability, but it's almost certainly going to be multiple batches in terms of how it's generated under good manufacturing conditions, GMP. Neural stem cells, like tissue-derived neural stem cells like we work with, have an additional challenge in that they may not be super stable over time and passage. We don't understand at the basic science level all of the things that regulate that super well. And almost certainly to do a large-scale clinical trial with a lot of patients, you may be looking at multiple batches and lines. So how do you show that comparability, not just to the FDA, because I can show that cell lines are similar. I'm going to show you in a minute. How do you really know that they're going to behave the same way, not just even in an animal model, but in an individual when you transplant them? And then, of course, we have mesenchymal cells on top of that, which are a whole nother game, almost certainly going to be multiple batches and lines. But I include it here because it influences, based on what we know from historical clinical trials with mesenchymal cells, or I think it should influence how we look at these things. The added issue is what happens when we try and leave the lab or sort of research process and scale up into clinical cell lines behind the walls of the GMP and trying to make everything match over time for how do we go forward. Many complications with that include the reagents that are used, the stability of whether or not you can get those reagents from suppliers, and just the physical difference that happen when you're handling cells in a laboratory versus behind GMP. So that adds up to a lot of potential for donor variation and batch-to-batch variation over time. And as I said, that means that the FDA is really looking for in vitro surrogate assays. They don't require in vivo validation of product, although we have argued in reviews and other places that that really should be a step that is required. And I can give you an example of this. So Osiris used a mesenchymal cell product that was designed to be a treatment for graft versus host disease, had tremendous efficacy in small-scale phase 1, phase 2 trials. When they went to their phase 3, it was kind of a resounding failure. And after that, some years, a number of people, Jacques Galipeau, for example, did a lot of work on this, came back and said it has to do with how you did your scale up. These cells are not stable across passage over time. And again, just what I said, we don't understand all the factors that are contributing to that. We don't have a good way to be able to evaluate in a dish or with in vitro measures alone. So that tells us, I think, that in vitro cellular and biochemical data is useful, but it can't be the end-all and be-all, and here's a place that we need to focus. So here was my lab in around 2017, where we had this tremendous leap forward for the field. We were so excited. Lots of people were excited. This colossal step backwards where we felt like, we just didn't even know what to do, right? With having to pull back from advocating a therapy that we had been really enthusiastic about. And for the trainees in the room, I thought very seriously of never touching a stem cell again so long as I lived, right? For the trainees in my lab, it's really hard, right? Because when you're doing translational research, you have to find basic biology for your trainees to be successful. You can't be plunging. We're not companies in academia. You can't be plunging only down the line of what's going to move you forward into the clinic. You have to balance with making sure your students are going to get dissertations and be able to publish and go on with their careers. And it's hard to do that when you're on this translational path. I was really lucky for the stem cell advocates in the room that I worked very closely with a number of spinal cord injury foundations. And I had a lot of contact over the years with folks that were living with these injuries and their families. And I found it really hard to walk away from this therapy knowing that people in the fields really wanted to have this, wanted to have the option to see that hope and the possibility of moving forward. So after much soul searching within this group, and very fortunately for us, some philanthropy that came from this gentleman, Mike Johnson here, we were able to retool and dug in and said, okay, we're going to make our own cell lines. We're going to keep control of them. We're going to figure out how they work, and we're going to try and take the next step forward towards translation. Later on in that process came an additional set of CIRM funding in the form of a DISC-2 and a TRAN grant, which we're currently working on. So as we did that, we realized we had to start thinking about even more than we had been already, the real basic science, and step to the side of that translational path and understand what was going on. So one level of question has to do with stem cells and how they were engrafting in the in vivo environment and behaving, which may be different than what they do in a dish. And so the concept here is that every cell in your body, and certainly a stem cell, has a niche. I didn't do it. There we go. Your basic niche and your basic cell illustrated here. But in many, certainly neurological diseases and neurological injuries, like spinal cord injury that I work in, seriously, I didn't touch anything. That niche can vary or change, right? So it might be normal when you start out, but in various states of disrepair or re-repair after the course of an injury. And a cell that's transplanted at different times is going to see different pieces in that niche, and we don't understand very well all of the factors that are interacting there. I think additional to that is that we really shouldn't assume that two cell lines derived the same will have identical characteristics. We've generally assumed that as a field. I'll come back to that in a moment. But also that the cells themselves have intrinsic properties that are going to define them, even if they're in the same category of cells, right? Even if they're all neural stem cells, they are going to have intrinsic programming that is going to matter ultimately in terms of what their function is. So we have to imagine that there's going to be extrinsic factors that are around them when we transplant them and intrinsic factors that are going to influence them when we transplant them. And that's going to have an effect on their maintenance, their self-renewal, their survival, their lineage restriction or not, their capacity to differentiate along different lineages or to different fates or not, and their ability to migrate. And for different injuries and diseases, I'm going to do this. That may be really important, right? And not important necessarily in the same way for spinal cord injury as for traumatic brain injury or stroke or multiple sclerosis. So understanding why stem cells behave in specific ways is really important for both basic science, because we should know that stuff, and also for clinical translation. And it's possible to kind of try and harmonize those things. It's possible to kind of try and harmonize those things. I can't see my slides, so there we go. By asking basic science and translational questions at the same time. So, sorry, I have to look over there because I can't see anything over here. So I want to give you an example of that. And these are 3 different spinal cords that have received thoracic level injuries and stem cell transplants at 3 different times. So either immediately after injury on the top panel, mid-career, 9 days post-injury, what we call subacute, or 30 days post-injury. Everything you see in brown is a human neural stem cell, and you can just look at these and say they're not behaving the same way. They were transplanted in the same place relative to the injury epicenter, so into the parenchyma above and below, but some cells are getting recruited in, and some cells are migrating away, and if you quantify up how many cells are there, it's the same number, but where they migrate to and what they become is different. And that dictates whether or not these cells, in this case of spinal cord injury, have the capacity for repair. So acute, those cells fail. Subacute and more chronic, those cells succeed in terms of yielding functional recovery. And so just understanding that basic idea that the cells are going to see a different niche and that's going to impact what their capacity is raises our level of complication, right? Because at least in the case of traumatic CNS injury, really no 2 injuries are the same. And so that's going to complicate our path to translation tremendously. Wow. Okay. So that's one aspect of basic science questions. And actually a big chunk of my lab is all on the path of what are those differences? What are cells listening to? How are they being instructed in the microenvironment? The second question that I raised, though, is how different are different human neural stem cell lines? And when we started to ask this question, after our colossal step back in terms of moving down the translational path, we really had no idea because there were very few of these lines. And for obvious reasons, these experiments are difficult. And so you tend to pick a line and transplant it and go with that. It's very difficult to compare multiple different lines across time. I don't know. All right, I'm going to go here because I can't see my slides. So we took that down the path of trying to establish how different it might be, just to ask the question, how different neural stem cell lines might be. We developed with some philanthropic funds a way to do a GMP-compliant method of isolating human neural stem cells from fetal tissue. We actually made 10 different lines, and we set out to characterize them in vitro and in vivo. And without going through the details, I'm just going to say these are all graphed as confetti bars, because what I want you to come away with is it's confetti in terms of how they behave. They have very different propensities for differentiation, for migration, for everything that you can measure in a dish. No two cell lines behaved exactly the same way. So to some degree, we got a little bit lucky with the initial line that we pursued in our partnership with the company on the clinical side because it worked. But the reality is that that confetti held true in vivo also, and only about 50% of the cell lines that we made showed efficacy in a spinal cord injury model. So they're all over the place in terms of their potential for repair. And fundamentally, I think many of you probably have the same variation and are unaware of it. And that's really important in terms of thinking about what you're doing in the lab, whether it's going to move forward on a translational basis or not. So can we narrow that down a little bit? In the path towards translation, that's called process development. And what we sought to do was to just ask whether we could get to a better place. Our original collaborators, founders of the company, actually gave us positive and negative control lines that we could pair with the lines that we had made. And then we went ahead and said, okay, can we look at interline variation as a tool to be able to understand factors that are going to give success or failure when you go to transplantation? The way we did that was to do a bunch of deep RNA-seq. We had transplanted ahead of time all these different lines, so we knew which ones gave efficacy and which ones failed. And we used that to develop a 24-gene profile that we felt might be predictive of in vivo efficacy. Then we generated 4 additional lines under GMP-compliant conditions. So our protocol behind a GMP facility, and we asked which ones of these were capable of working or not prospectively using the RNA-seq profile that we had generated, and we predicted, according to our 50% rule, that 2 would succeed and 2 would fail. And actually, when we went ahead and did the actual in vivo experiment, that's exactly what happened. So that suggests that it may be possible to use some more complex methods to predict success and failure of the different cell lines across batches, across lines, that could be useful in terms of neural stem cells. And that's what we're hoping is going to help us move forward in our next translational stage. Critical also when you're thinking about this translational path and how do you move forward, in this case, for spinal cord injury in our story, is identifying quality attributes, critical quality attributes of the cell. This is a place where, coming back to what had been done before with mesenchymal cells, it was really quite challenging that one of the gaps in this early OSIRIS trial was that hadn't been done. So they didn't have any features to look at to say these cells have retained their identity, and so we expect them to work in this phase 3 trial as opposed to not work. So we tested a number of different parameters, including passage number and seeding density, right, as a start towards process development. Why would we do that? Well, you can see here that if you look at the max final products that can be made, depending upon if you're starting at the highest seeding density and the lowest passage, in fact, is maybe 9 or 10,000 vials. But if you go to the lowest seeding density and lowest starting passage, that might be 500,000 vials, right? It's exponential in terms of what you can get. So you really want to get to max possible production. So we tested all of these different things in a dish. I'm not going to walk through the details of all of these. We applied our RNA-seq profile here, and we found that each one of these clustered super close together, predicting that they were all going to work. Yippee-ki-yay. But they actually don't. In fact, only one of the 2 conditions that we went forward into in vivo testing worked, and I couldn't tell you why. It isn't anything to do with what we would have predicted as a critical quality attribute, CD133 expression and so on, and it isn't what we have been super excited about in terms of our RNA-seq profile. And I show that to you, again, 1 step forward, 1 step back, a couple of steps sideways, because, 1, that's just the game in terms of moving forward, in terms of translation. And 2, I think it's really important for us to start recognizing that as a field, and that we may not, for the near future, get to really good in vitro data predictions. We may need to just build in the expense and the time of understanding not just the basic science, but also doing validation in an in vivo setting to be able to move forward. Okay, so I wanted to just give an overall sense here. All of this has to be done with a target product profile in mind and a path to GMP scale up in production. I'm not going to walk through this. This is what a target product profile looks like. They get pretty in depth in terms of thinking about what patient population, how are you going to stratify, what are you going to measure, what are you going to know about your cells. And then, of course, there's release testing parameters from a safety perspective. There's also an issue of in a field, like if I'm going to a neurosurgeon and saying, hey, will you transplant my cells? In a field, there's an issue of how much is enough, right? When is it justified to move forward into a clinical trial versus not? And I want to give you a sense of what that looks like in terms of these cells. So this overall chart is every cell that's been transplanted into a chronic cervical injury model and published, and what the effect size was of those. The two at the top are the cell line that we're trying to move forward that we've developed, UCI 191, which have a pretty wonkin effect size. So that's very promising in terms of being able to move forward. I would argue that if you have an effect size that's much less than that in an animal model, it's probably not the place to think about moving forward in a responsible way towards clinical trials. And then, of course, beyond that, how much do you have to replicate? Like, how many times should you use these cells in a dish in different animal models to know that nothing bad happens? You don't induce pain or allodynia or other problems versus getting a positive effect in terms of your cells. And again, I just want to give you a sense of what that looks like in terms of this translational path just within my lab, right? It's a lot of studies that you end up doing. And it's a lot of studies where you're not going after the basic science, you're just going after safety and efficacy. So here's where we are. We have navigated through selecting our primary cell line. We actually have done GMP manufacture of that cell line. It's got a few hiccups, and we're in process right now with the in vivo testing for that cell line to understand if we have a final product that can meet all the criteria that we've established to be able to move forward. And I tell you that because I think this is a really important mission that CIRM has set out. I think there's a tremendous amount of basic science that is really important and buried in here. I think we have to be funding along that full spectrum, from one step forward to back to understanding the basic science to one step side on to the next thing, because this is a rinse and repeat process in order to get to success. A few challenges that are certainly remaining, and I like to show this slide because, you know, we work mainly in mouse models, and that's a human brain and spinal cord, so scale is obviously an issue. I think a critical issue that we're going to be facing in the field, we're certainly working on with one set of our projects, is for a cell population like I'm talking about, where we know we have to have long-term engraftment of those cells, how do you really get that? Do you want to keep people with acute central nervous system injuries on chronic immunosuppression for the rest of their lives when they already have challenges? And so a way to be able to get around immunotolerance or developing immunoinvisible cells is obviously something that's quite important. And then there's just the practical clinical side. You have to work very closely with clinicians over patient stratification and clinical trial design and what makes sense, an entire sphere of things that's outside the laboratory in terms of what you do. So with that, hopefully I'm not too far over. A bunch of shout outs here. Of course, CIRM is a funding agency along with the National Institute of Health, Christopher and Dana Reeve Foundation, without whom I probably would have abandoned stem cells, and Wings for Life, and some really critical folks up here, particularly Katja Pilti, who developed this protocol in our lab and generated all of the lines that I talked about today, and also Brian Cummings, who's on faculty at UCI and has been a collaborator throughout these projects. So thank you for your attention.
Hello, my name is Dennis. I have a couple questions. I mean, some of them you've already answered them, but as a STEM scientist, you've mentioned some of the challenges. What are some of the challenges the scientists should watch out for?
So I can understand why the previous speakers had trouble. It's echoey up here. So the question is, what are some of the challenges that stem cell scientists or scientists should watch out for? So I'll frame this from speaking to the trainees that are here perspective. And I think a challenge there is translation is exciting and the impact is, it's thrilling to think about being able to have an impact that will help people. But that's a hard road, and it's not something that you're going to accomplish in an internship or a dissertation, right, or probably your postdoc. We're talking about work that spans over 20 years at this point, right? And so think about having multiple options on deck simultaneously always. You should always have a sure thing project. You should always have something that's aspirational. And I think the challenge is sustaining the energy to do both at the same time.
Okay, thank you. And then you talked about earlier on in your presentation, you talked about passages up to 20. And then some of the last slide decks, I saw up to passage 7. What's the highest number of passages you can reach to?
Yeah. So the question is, what's the highest number of passages we can go with the cell line? And I don't know. So early on in neural stem cell work, I mean, we're going back a long way for this type of cell population. There are a number of papers published that suggesting that after around P15 or P20, a multipotent cell that is unbiased in making neurons, oligodendrocytes, astrocytes would become glialized. And we never wanted that early on when we started out with these projects. And that's because we don't generally think about getting a bunch of astrocytes in the spinal cord after transplantation as being something that would be beneficial in terms of repair. So we were always very cautious in terms of our passage number. We never really have used cells in the lab that are over P10. That said, we've never tested beyond that in terms of in vivo transplantation. And it's a really important question on the basic science side of things, right? What's changing? Can you stop it from changing just by virtue of how you culture the cells so that now you could get cells that could go really long time? And how much does it matter when you transplant into an in vivo environment? So that's the space, I think, where basic science and translational science really mesh well, because there's certainly great basic biology that's buried in there, and I don't know the answer.
All right, thank you. That's all my questions.
Hi, thank you for a very interesting talk. I have a question from a regulatory standpoint. From your past experience and throughout all these years, how did you see the interactions you had with FDA evolved, and how was your how was the evolution of the, because I know they've changed a lot and they are really more open to dialogue, and I know this is very important from the safety perspective and regulatory standpoint. So if you have any comments on that.
So I think the FDA, in my somewhat limited interactions, right? I think the FDA gets a bad rap. So I hear a lot of folks at companies kind of jump up and down and rant and rave about working with the FDA and how they won't consider our options and how they're very narrow in terms of their interpretation. One, I think that's their job because their primary issue is safety, right? And this is a very unknown, unregulated domain for how to go forward. In my personal interactions with regulatory agencies, I found them to be incredibly reasonable. I never had a situation where I said, well, I think we should do this because it will support, this data, and that will tell us what we need to know in terms of going forward into a set of clinical subjects. I've never felt like they were not open to that discussion or reasonable. I think the challenge where that reputation comes out is you don't often, like a very small number of your interactions with them, do you actually get on the phone or sit in a room with anyone, right? And in fact, it's only in the old days when we were doing these first studies where you even get to sit in a room with them, right? Where it's a dialogue. It's mostly a very stilted framing of questions. Do you agree that if this, then that? And you tend to get somewhat cryptic answers back because you're not able to have a dialogue, right? And that creates uncertainty, which is hard to deal with when you're trying to move things forward as a translational scientist in a lab. And it's really challenging for companies, right? Because their bottom line is always looming. So I think it's not that they're unreasonable, it's that the dialogue is stilted. And if there's anything that would help us to get past that, it would be doing a better job of that. I don't know what the solution would be.
All right, let's thank Eileen again.